Namaste. Today I am going to perform the dissection of a brightly colored flower called Hibiscus. The scientific name is Rosa sinensis. It is a complete flower because it has all the four whorls. The outermost whorl is called the apicalyx. They may be 5 to 8 in number. They are finger-like projections. You can remove it with your fingers one by one and can display it afterwards. So I am removing the apicalyx. Now the second whorl is the calyx, green in color. So in order to remove the calyx, you have to make a small slit from downwards to upwards, just a cut, and you remove it with your fingers slowly. The calyx consists of sepals. You will see there are 5 sepals, 1, 2, 3, 4, 5, and they are united, so they are called gamosepalous. Now there are 5 petals. They are also 5 in number, but they are not united, they are free, so they are called polypetalous. So remove one by one all the petals. Now you can see there is a androecium. Can you see? Here there are stamens are there. The androecium is surrounding the gynoecium. So in order to remove the androecium, you have to make a thin cut like this. You have to loosen the staminal tube. It is very sticky. Now slowly remove all the remaining androecium. Now you can see the gynoecium part. This is the ovary, white style and 5 stigma. Can you see? Now I'll display all these whorls and label it. First I will display the apicalyx with a pin. So these are apicalyx displayed. They are 5 to 8 in number depending upon the flower. These are sepals. And petals I'll arrange like a flower. Now I label all the whorls. These are sepals and they are gamosepalous. These are petals. They are polypetalous. These are all stamens. And this is the staminal tube. These are 5 stigma. Style, and this is the ovary. This is the pedicel. So I have done the practical. So in this way you have to dissect the hibiscus flower. Thank you everyone.